In this section, we will talk about acetylcholine and cholinergic transmission. Now, let's discuss acetylcholine synthesis. This is presynaptic neuron, while this is postsynaptic neuron. Choline is transported inside the neuron by energy and sodium-dependent transport system. Acetyl coenzyme A is synthesized in mitochondria. The reaction of coenzyme A and choline is catalyzed by choline acetyltransferase. Acetylcholine is then transferred to presynaptic vesicle to protect from degradation. Vesicles on presynaptic nerve terminals empty their contents into the synaptic cleft. When the cytosolic calcium concentration rises in response to incoming action potentials, acetylcholine is then released and bind to postsynaptic receptors and leads to cholinergic response. But epinephrine and norepinephrine can inhibit acetylcholine release by stimulating presynaptic alpha-2 adrenoceptors. Acetylcholine blocks its own release by binding to presynaptic autoreceptors, which gives a negative feedback, thus signals to stop the further synthesis of acetylcholine. Now, an enzyme called as acetylcholinesterase at synaptic cleft breaks acetylcholine into acetate and choline. This choline is taken up again by presynaptic neuron, and the process for synthesis of acetylcholine starts again. Now let's discuss the types of cholinergic receptors. As we have discussed that, there are two types of cholinergic receptors. First is muscarinic receptor, which has high affinity for muscarine. And second is nicotinic receptors, which has high affinity for nicotine. There are 5 types of muscarinic receptors: M1, M2, M3, M4, and M5. But in autonomic nervous system, we will only talk about M1, M2, and M3. M1 occurs mainly on autonomic ganglia, CNS, and exocrine gland cells. M2 occurs in the heart. M3 occurs mainly in smooth muscles. M1, M2, and M3 are metabotropic receptors. They indirectly affect synaptic transmission through G proteins. Let's understand this. When acetylcholine binds cholinergic M1 receptors, they activate phospholipase C-beta through GQ proteins in the postganglionic neuron. And inositol triphosphate and diacylglycerol are released as second messengers that stimulate calcium influx and a late excitatory postsynaptic potential. In heart, M2 functions mainly through GI protein. The GI protein opens specific potassium channels located mainly in the sinoatrial node, atrioventricular node, and atrial cells, thereby exerting negative chronotropic and dromotropic effects on the heart. The GI protein also inhibits adenylyl cyclase, thereby reducing calcium influx. M3 are similar to M1 cholineceptors. M3 cholineceptors trigger contractions by stimulating calcium influx. However, they can also induce relaxation by activating calcium-dependent nitric oxide synthase, for example, in endothelial cells. Now, 2 types of nicotinic receptors are there: NM and NN. NM is located in the neuromuscular junction, which causes the contraction of skeletal muscles by way of end plate potential. NN causes depolarization in autonomic ganglia, resulting in postganglionic impulse. Nerve-specific NN cholineceptors on autonomic ganglia differ from muscle-specific NM cholineceptors on motor end plates, in that they are formed by different subunits. They are similar in that they are both ionotropic receptors, means they act as cholinergic and cation channels at the same time. When acetylcholine binds to N-nicotinic receptors, leads to rapid sodium and calcium influx and early or rapid excitatory postsynaptic potentials, which trigger postsynaptic action potentials once they rise above threshold, while efflux of potassium also occurs. Now let's understand the cholinergic transmission. This is preganglionic neuron. This is postganglionic neuron. As we have discussed that vesicles on presynaptic nerve terminals empty their contents into the synaptic cleft when the cytosolic calcium concentration rises in response to incoming action potentials. Epinephrine and norepinephrine can inhibit acetylcholine release by stimulating presynaptic alpha-2 adrenoceptors. In postganglionic parasympathetic fibers, acetylcholine blocks its own release by binding to presynaptic autoreceptors. This has already been discussed. Acetylcholine binds to postsynaptic cholinergic receptors, or cholineceptors, in autonomic ganglia and organs innervated by parasympathetic fibers, as in the heart, smooth muscles such as the eye, bronchi, ureter, bladder, genitals, blood vessels, esophagus, and gastrointestinal tract, salivary glands, lacrimal glands, and sympathetically innervated sweat glands. Cholineceptors are nicotinic or muscarinic, and cholineceptors, which are nicotinic, can be stimulated by the alkaloid nicotine, whereas cholineceptors, which are muscarinic, can be stimulated by the alkaloid mushroom poison muscarine. It has also been discussed. Nerve-specific N-cholineceptors on autonomic ganglia differ from muscle-specific N-cholineceptors on motor end plates in that they are formed by different subunits. They are similar in that they are both ionotropic receptors, means they act as cholineceptors and cation channels at the same time. Acetylcholine binding leads to rapid sodium and calcium influx and potassium efflux and in early or rapid excitatory postsynaptic potentials, which trigger postsynaptic action potentials once they rise above threshold at 20 milliseconds. M-cholineceptor, M1, M2, and M3 are metabotropic receptors. They indirectly affect synaptic transmission through G proteins. M1 cholineceptors occur mainly on autonomic ganglia, CNS, and exocrine gland cells. They activate phospholipase C-beta through GQ protein in the postganglionic neuron, and inositol triphosphate and diacylglycerol are released as second messengers that stimulate calcium influx and a late excitatory postsynaptic potential at 2 seconds. Synaptic signal transmission is modulated by the late excitatory postsynaptic potential as well as by co-transmitting peptides. They are released and then bind to peptide receptor that trigger peptidergic excitatory postsynaptic potential or inhibitory postsynaptic potentials at 60 seconds. Now let's understand cholinergic transmission in heart through M2 receptor. This is preganglion parasympathetic neuron. This is SA node or AV node cell. As we have discussed that vesicles on presynaptic nerve terminals empty their contents into the synaptic cleft when the cytosolic calcium concentration rises in response to incoming action potentials. In postganglionic parasympathetic fibers, acetylcholine blocks its own release by binding to presynaptic autoreceptors and produced negative feedback to stop acetylcholine production. Acetylcholine binds to postsynaptic cholinergic receptors, M2. In heart, M2 function mainly through GI protein. The GI protein opens specific potassium channels located mainly in the sinoatrial node, atrioventricular node, and atrial cells, causing hyperpolarization, thereby exerting negative chronotropic effects from chrono, meaning time, and tropos, meaning a turn, so decrease in heart rate through sinoatrial node, and negative dromotropic effects on the heart, means decrease in rate of conduction through AV node. The GI protein also inhibits adenylyl cyclase as decreased conversion of CAMP from ATP, decreased protein kinase A, and thereby reducing calcium influx. Termination of acetylcholine action is achieved by acetylcholinesterase-mediated cleavage of acetylcholine molecules in the synaptic cleft. Approximately 50% of the liberated choline is reabsorbed by presynaptic nerve endings. This was all about acetylcholine and cholinergic transmission. In our next section, we will talk about the catecholamines, adrenergic transmission, and adrenoceptors.